Welcome to This Explainer. Today, we're looking at what is arguably the most remarkable and totally verifiable claim in modern neuroscience. We're going to explore how psilocybin doesn't just, change how you think or temporarily shift your mood, but how it physically rebuilds the structural basis of your cognition. If you're fascinated by neuroplasticity and the biological mechanisms of the mind, well, you are in exactly the right place. So get ready, because we are looking at a literal architectural paradigm shift in the human brain.
Okay, let's dive right into this. For decades, the field of psychiatry has really leaned on this chemical imbalance model. We've basically been told that mood disorders are simply a matter of having too little or too much of certain neurotransmitters floating around in our heads. But that outdated model is being aggressively challenged right now by the neurotrophic hypothesis of depression. The science is asking a much, much deeper question. Is depression just a feeling or maybe a temporary chemical deficit? Well, the evidence strongly suggests it's not. Actually, it's the result of impaired neuroplasticity. It's a fundamental loss of your brain's ability to physically adapt, rewire, and grow new connections. To put just how physical this is in a perspective, look at the staggering number: 30%. Research spanning all the way from 1996 to 2019 has consistently shown a reduction in hippocampal volume by up to 30% in patients dealing with recurrent major depression. I mean, let that really sink in for a second. Chronic stress, which we know is the primary environmental trigger for depression, causes actual stress-induced neuronal atrophy. The hippocampus literally physically shrinks. So depression isn't just some abstract emotional state. It's a profound structural condition. You literally have fewer connections and smaller neural pathways. To truly treat it, we can't just manage the symptoms, right? We have to rebuild that lost architecture.
And that brings us to the real hero of our story today. Now, it sounds like something you'd read on the back of a fancy supplement bottle, but brain-derived neurotrophic factor, or BDNF for short, is basically the master builder of your brain. It is the absolute primary protein responsible for your brain's ability to rebuild itself. It's highly concentrated in the hippocampus and the prefrontal cortex, and BDNF is essentially the biological signal that makes structural growth possible. So whenever you're learning something new, adapting to a situation, or healing, BDNF is the agent in there doing the heavy lifting.
When BDNF binds to a specific receptor, it's called TRKB, it doesn't just send a quiet little chemical whisper. No, it sends a direct biological command to the neuron saying, grow, connect, strengthen. It promotes the survival of neurons when they're under metabolic stress. It stimulates the birth of entirely new functional neurons. And crucially, it increases the density of what are called dendritic spines. Think of these spines as the actual physical platforms where all your synaptic communication happens.
And this brilliantly illustrates the massive paradigm shift we're talking about today. So traditional antidepressants, like SSRIs, they work by increasing synaptic serotonin, which then gradually upregulates that BDNF expression over 2 to 4 weeks of daily dosing. The serotonin is really just a messenger in this process, and let's face it, it's a frustratingly slow one. But psilocybin? It completely bypasses this weeks-long delay. When psilocin binds to the 5-HT2A receptors, it simultaneously triggers rapid BDNF expression on a timescale of hours, not weeks. Recent research even suggests psychedelics bind to that TRKB receptor with unusually high affinity, meaning they engage the neuroplasticity pathway directly. The results of this direct engagement are honestly absolutely explosive. There was a landmark 2021 study out of Yale University from the Schau Laboratory, and they actually tracked individual dendritic spines in living animals. Following just a single dose of psilocybin, they observed a 12-fold increase in dendritic spine density in frontal cortex neurons within just 24 hours. 12 times the density. This represents a massive, incredibly rapid expansion of the brain's connective architecture. We're talking new pathways for processing information, new routes for communication, and entirely new structural options for behavior and cognition, all springing up almost overnight. Now, this leads us to what I think is the most mind-bending part of all the psilocybin research. Pharmacologically speaking, psilocybin is completely metabolized and eliminated from your body within about 6 to 8 hours. Yet the clinical effects and those profound improvements in mood can persist for 6 to 12 months. So how is that even possible? Well, because, as the data shows, the brain literally rebuilds itself. Those new dendritic spines they saw in the Yale study, they weren't just temporary blips. They were completely functional, showing increased spine head width, which is a sure sign of synaptic strength, and the vast majority of them persisted for over a month. The healing massively outlasts the drug's actual presence in your system because the physical hardware of the brain has been permanently upgraded. Researchers over at Imperial College London and Johns Hopkins have converged on this idea they call the therapeutic window hypothesis to explain exactly how this works. Think of it like a two-step home renovation. Step one is the demolition. The acute psychedelic experience disrupts what's called the default mode network, essentially your brain's rigid, everyday autopilot, and that dissolves the entrenched neural patterns that maintain depressive or pathological states. Then, step two is construction. During this newly opened window of flexibility, that rapid BDNF expression physically constructs the replacement pathways. Both phases are absolutely necessary here. The demolition clears the site, and the BDNF construction builds the brand new architecture. So the absolutely crucial point here is what you actually do during this construction phase. The brain is in this highly unusual state of structural malleability. It is hyperplastic. And this perfectly explains why those concepts of set, setting, and integration matter so profoundly in psychedelic therapy. The behaviors, the thoughts, and the therapeutic framing that you engage in during this malleable window, they are preferentially encoded directly into your new neural architecture. Positive integration doesn't just make you feel good emotionally. It literally dictates the physical shape and the strength of the new synapses your brain is actively building. All right, let's move on and look at sustaining this new architecture with what we call the BDNF ecosystem. Because, BDNF expression isn't just exclusive to psilocybin, and this ecosystem gives you immense agency over your own neuroplasticity. If we view psilocybin as the powerful catalyst that kicks off the reconstruction, we then really have to look at what sustains that growth day to day. First up, aerobic exercise. It is the most potent natural BDNF upregulator we know of. It gets that cerebral blood flow going and stimulates BDNF transcription in the hippocampus in just 20 to 30 minutes. Second, your diet plays a huge role. And yes, ceremonial cacao is the exact scientific justification you've been looking for to eat way more dark chocolate. The flavonols in it synergistically increase BDNF levels. And finally, slow-wave sleep. Listen, this one is non-negotiable. Sleep is when the structural consolidation actually happens. During deep sleep, all those new synaptic connections you've built are either stabilized or pruned away. Without proper sleep, BDNF expression is completely suppressed and the rebuilding just halts. What we're looking at here is nothing short of the structural future of psychiatry. We are finally, thankfully, moving past the era of mere neurotransmitter management. The days where we just continuously adjusted chemical messengers to get temporary relief without ever actually changing the neurons themselves. We are entering a real era of verifiable, measurable neural rebuilding. Dendritic spines can literally be counted under a microscope now. Hippocampal volume can be accurately measured with an MRI. The science proves, without a doubt, that your brain is not fixed. It is not a static machine. It is a dynamic, living architecture that responds incredibly well to the right molecular signals. And that is really the ultimate takeaway from this explainer today. Psilocybin might be the most potent known trigger we have for initiating this massive architectural upgrade. But the ongoing construction is deeply, deeply intertwined with how you live, how you move, and how you sleep. You have the structural flexibility and the biological tools to rewrite the very basis of your cognition. So I'm going to leave you with this one final thought. Your brain is a living architecture, capable of brilliant, beautiful neuroplasticity. What will you build with it? Thanks so much for learning with me today.